tea. The milkman, the paper boy, evening TV. Did I get delivered here? Somebody tell me, please. This old world confusing me. Clouds as mean as you've ever seen. Ain't a bird who knows you're true. Then a little voice inside you whispers, don't sell your
Alright, this is the sweep that we've done for macroinvertebrates. We sweeped along the surface and by the bottom, and this is what we picked up.
Step one, we just did the thing.
Yeah, so the thing is a bunch of chemical tests. Thanks, Lance.
It's sweep. The wind is 14 miles an hour coming from the south. It is 13 miles an hour coming from the south. 72 degrees. It's March 15th, 16th. It's 11:04 o'clock. Nate is going to go in the water. And he's going to get half a bucket. He's going to go in there, testing it scientifically.
The big rock?
Not a big rock. This is big.
Take your shoes off, bro.
I don't know if you guys can see this on film. We found a big rock. Nate is going to go in there and get it.
Fill it up.
Oh my God. Rinse it out.
Alright. Here's the rock here. It's gonna fit.
Now that we have the rock, we're going to science it.
Here we're looking for macroinvertebrates. This allows us to pick up macroinvertebrates that stick to the bottoms of rocks and different debris like wood, and then test them and see how the level, or see what the level of pollution may be in the lake. So here's more debris. This is a chunk of what looks to be the outermost layer of what appears to be bark. And here you can see a mayfly larva, which you can see that there's mayfly larvae on this. There are a lot of other macroinvertebrates as well on this debris.
So, but we got a clam or a mussel in there.
That's our water.
This is the E. coli test. So we have a gel that is in liquid form right now. We're going to take 3 milliliters of lake water and mix it with the gel, and the gel is going to allow whatever coliform is picked up.
So that's 3 milliliters into the E. coli scan. And now we put it in this petri dish and it's going to solidify.
In the water to grow and feed, and then we'll see it later, like right here. No, that wasn't right there, but you know. So here we're doing different chemical tests for the lake. We have a nitrate test, a phosphate test, and a pH test. These are going to test different levels of phosphate and nitrate in the water, which can tell us if there's a possibility for an algae bloom, and then also what the acidity of our lake is.
Okay, so we did our pH, nitrate, and phosphate tests at Lake Max. For phosphate, we got 2 ppm. For nitrate, we got 5 ppm. And then for our pH, we got a 7. coliform test?
So this is what we have for Culver. Right now we are 53 degrees.
We already did that.
0% chance of
That's
rain.
not our job today.
What's the temp?
The temperature is 53 degrees right now.
Is there a water temp on there?
The water temp does not. It feels like 49. That's all I gotta say.
We're doing dissolved oxygen.
Try to rinse that out.
This is a pH test. Sets are doing phosphate and nitrate tests. Yep, about. And then after that, we're gonna get the results and let you know why we did this.
Okay.
The first tablet didn't really work that well, so we're trying it again with the second tablet. That was a lot easier, and I think that I didn't use the right tab the first time. Go ahead and redo this test. Of course, on the first one, and put the wrong fit tab in. So we're going to redo this with the right tab. And as you can see, it pretty much like immediately changes color. So that's different.
Yep.
I'm not surprised.
No, you just like collect the water. There's like a Well, they're just making it way worse now.
I feel like it's gonna be like 0.
They're just making it way worse now.
You got a rock.
You got a rock. So, scale from 1 to 10.
How dead?
There's a something. It's dead, but
There is a something.
there's a body.
It's a body.
Body hit the ground. That, or is that just might just be
That's
wood?
wood. So that's our found wood. All right. I think we're giving up.
All the way in and then cap it in the water.
Oh, it's in the boot. It's in the boot.
All right, we got it. I thought there was geese. Doing this all oxygen test now. Step 1.
Cue the montage music.
One eternity later.
The water temperature.
So here we are taking the temperature of the water, and it comes out to be 14.4 degrees Celsius. I didn't say it. Lance is now preparing the phosphate, nitrate, and pH tests so we can do that. Alright, so this is a little recap on the bus ride back, and Lance is going to go over some of the temperatures and wind speeds and such.
52 degrees. Wind speed was about 15 miles an hour. We took care of all of our pH measurements and things like that. So we did phosphate count, nitrogen count, and our pH. So we have all those stats written down. Again, wind speed was 50 miles an hour. It was 52 degrees. Day 4, we are at the outlet. We're going to be taking some samples, doing our tests, and we're doing the E. coli test as well today. Water feels great on my boots, so let's get after it today. Probably Lance's good. Well, the tallness doesn't really factor into the boot. All right. Cool. It's hot. In this hose. So. We got the water. Wait, it's like an inch. It's like half an inch.
Unsuccessful. Snails. A family.
Boom.
Yeah.
They're getting a water test right now. We got a tadpole, so we have that in our macro bucket. And then we're gonna run these tests.
You want to tell them water temp? Water temp is 19.6 degrees Celsius.
We're done with everything else. Wait.
That's tadpole. Any movement, I'll put the net on it. Just keep walking this way with me.
I'm just gonna point my arm towards. Oh, there's one over there. Where? It's right there. Did it jump in?
I don't know. Get it? Now, oh my god, I got it!
You got it?
A frog. I just blind caught a frog. I'm a legend. No!
No!
It's gone forever.
Scoop
Did you get that on video? Okay. We get all the tests done?
We just have to do this one. Let's go. Of the macro test that we did today. There's a mayfly skin shed. Oh Lord. Mayfly skin shed. There were a couple.
Tadpole.
There's a tadpole. So frog. There were a couple of shells, left and right-handed. Aquatic plants. There's a lot of them. But I'm not seeing any other. No, there's a little worm there. That looks like. What would that be? Looks like a midge larva.
Where's the tadpole?
Tadpole's over here.
Oh, I see you.
Yeah.
Should empty some of the water out.
Yep.
Science. Oh god.
But like here, there was one. Might have been this. There's a new log ground which is shown tonight, and he went to design a restaurant.
Okay, we made it on the bus. We're really late. It's at turn 8 and I had to change. We were out of pens. Not really. Science lab.
Yeah. And we ran.
I'm sweating. Day 5.
Let's Hey, you know what I'm thinking? I'm thinking we just go in it. We go in it. Let's do it.
Nitrate's 5.
Ten? We're good. Houston, we're safe. 10 for both. All right. We're testing nitrogen, nitrate, oxygen, and yep, in the tubes. We're testing the nitrate. We have 5 milliliters of the water sample here. We just put our tablet in. We just gotta wait for 4 to 5 minutes and test the color out.
Where you going? Oh, okay, I was gonna do that, sorry.
Looks like that could either be a midge larvae or probably a riffle beetle. Yeah, it's probably a riffle beetle.
Nato, can I see your camera?
Yeah.
Oh, and then there's something right there as well.
Alright.
What is that? It's got. How many does it have? Does it have 3?
So it'd be a good spot to get some like uber nature-y B-roll.
Okay.
What? It'd be a good spot.
It's right there.
What is it?
Something. I don't know exactly what.
A bit.
Hey, wait, we got a blood larvae. You want to take a video of it? It's right there. You see that? Bloodworm something. Bloodworm.
Dead crayfish. Crawfish.
Yeah, I can't lift that.
Like a Nebo.
First question that we have is discussing the watershed in Lake Max. So our project revolved around the question of, is the wetland doing its job? And we looked at the watershed as an important piece in solving that question, because the water that comes in via the streams that feed into Lake Max, and also the wetland in this case, are feeding into the lake, and then that water stays there for 7 years. So it's a pretty good test as to see how the wetland is functioning and if it's actually doing its job properly. So we go in there and we do these different chemical tests, looking at phosphate, nitrates, the pH of the lake, things of that matter, to tell us if the wetland is actually providing a healthy, beneficial impact on the lake. Second question is talking about the wetland and how it works to clean the water. So the way that the wetland actually cleans the water is it filters it through allowing the water to stay in one area for some time before it goes on and stays in the lake for 7 years. And in doing that, it allows sediment to go through that may have different fertilizers with phosphates and nitrate nutrients in them, and allows them to go into the plants, the aquatic plants that we have there in the watershed, in the wetland, instead of going into the lake and creating algae blooms that kill off large populations of fish and other aquatic life around them and deplete the area of the dissolved oxygen.
So Lake Max is what is called a watershed, and watershed is, well, basically it collects water from different kind of rivers, streams, or ponds around that area and puts it into one giant lake or watershed. So Lake Maxinkuckee basically is collecting all the water from Cardinal Lake, every ponds around the Culver community, and even the waste that is produced in the town of Culver, and also even in our school of Culver Academies. And it puts into one giant mass of water, body of water in Indiana. So what it does is it leaves an opportunity for a lot of plants or animals to grow in the watershed, and it enables a lot of beneficial or even harmful effects of community, and then we can see if it is getting hurt towards because of the chemical waste that we are producing, or stuff that is beneficial that we can actually research about in our bio class. Fourth question is, the majority of the water comes from the drains into the lake, and the three main tributaries are the Curtis Ditch, Wilson Ditch, and the Klein Ditch. Those three ditches are located in each various positions, and we have also researched in Wilson Ditch and saw how the ditches work and how the process before the water reaches into Lake Max works, and how that system is started and where the water comes from. So the watershed is mainly rural, with the agricultural lake composing about 41% of watershed, and it contributes to drain into, and those three ditches drain into the lake and contribute approximately 70% of the phosphorus loading of that comprises in the Culver Lake of Max.
So one of the concerns that we have when assessing results from the various areas that we collected data at was that how are some external factors going to affect the results. And when we're looking at things like the location, area plays a very big part of that. So if there's a construction site by an area where we sampled something, there may be some type of contamination that wasn't there before. And that could also give us an idea of how the area is going to affect the results of the water and the contamination level, which would then affect the health of the lake in the long run. So at a couple of sites, there were areas where we saw a lot of junk and debris, human debris inside of the actual streams and rivers, and this can affect the health of the lake. Things like television sets being inside of the water, where there could be some corrosion through copper wire, or maybe some iron metals, and there could be an iron oxide consuming organism that affects the lake, which you can even see sometimes in some of the smaller outputs that we have going into the lake along the lakeside dorms. And the area is one of the main concerns when looking at that, because you can see how there are some external factors that could come into play in the contamination of the lake. Next, how was the Wilson Wetland constructed? Very simply to help the health of the lake. So there was a biologist that came in so many years ago when the lake was almost completely destroyed by algae and was almost unbearable as a vacation home, and also the health of the lake was extremely poor, and it was coming to the point where we may not even have Lake Max anymore. So he concluded that we needed to start to filter some of the bodies of water that were running in to Lake Max, and one of the ideas was to build a wetland. Wilson Wetland was the result of that. It was the first
wetland, man-made wetland in Indiana by Culver faculty, staff, students, and it helps to filter the water that goes into the lake and provides an extreme benefit of the actual health of the lake by filtering out some of those nutrients that we talked about before, sediments, things like that, that creates a positive effect on Lake Max.
We had 5 sampling sites. First, the lake, lake inlet, lake outlet, the Wilson Ditch inlet, and Wilson Ditch outlet. So we had to sample all those sampling sites in 5 days, in the span of 5 days. And what the results that we had was the lake had a medium quality of, the water quality was medium, lake inlet was bad, and lake outlet, Wilson Ditch inlet, and Wilson Ditch outlet was all medium. So what this tells us is that it's not that good and it's not that bad. So basically what it says in the medium is that there are some parts that are not filtered out, and there are some parts that are going out that is not very beneficial to the lake, but it's not that harmful to the environment around there. So that was what I got. That was what we are as a group got. But we believe that the lake inlet's water quality, which was bad, could have been much better because we had a storm the day before, and we believe that the nutrients and the sediments and fertilizers that were in the inlet and the wetland did not filter out for 7 days, did not stay in there for 7 days, so it had a hard time, and it overflowed and spread it out to the lake without getting filtered. And that is one of the problems that we had in the wetland, of how it is too small of a size compared with the size of the lake. And if the wetland is overflowed because of the storms or because of the rainfalls, especially during this time of the season, it does not do its job properly, and we would have this kind of a crooked number and crooked data that would really contrast with what we expected and contrast with the hypothesis that we had.